Let's learn about protein synthesis. Now, to learn about protein synthesis, we're going to locate the process in the parts of the cell where they take place. We're going to be locating the process inside the nucleus, which is here. This side of this membrane. So inside here, this is the nucleus, and this is the cytoplasm. In our nuclear structure, we have the nuclear membrane, outer and inner membrane, and we have the nuclear pores. Now, the nuclear pores only allow small molecules to move through. What we really want in protein synthesis, which happens in the ribosomes, which is this structure here, we need to get this specific base sequence to the ribosome so protein synthesis can take place. This happens in two stages, transcription and translation. Transcription happens inside the nucleus. Translation happens outside the nucleus. Now, in transcription, what we need to do is work out which gene it is that we want to transcribe. And DNA helicase is an enzyme that will identify the gene that we want to transcribe and break the hydrogen bonds that would have been between the between the complementary bases. So DNA helicase breaks the H bonds between complementary bases in the region of the DNA that we want to express. This could be the gene for insulin. Okay, this could be the gene for insulin that is being expressed in the cells in the pancreas. So only those bases are the H bonds between those specific bases are broken, leaving our bases now exposed. These bases are now exposed. Also inside the nucleoplasm, we have free-floating nucleotides. Okay, these nucleotides that can move, and they do move, and they will move to orientate themselves against their complementary bases. So this one would move there. G would then bind with C. C would come and bind with G. Okay, T is replaced by uracil in the production of mRNA, and so on. And what we have there is our exposed bases, free-floating nucleotides binding with the exposed bases, and those hydrogen bonds forming, just for a moment, just to keep them in that sequence, because it's the sequence that's so important, the sequence of these bases. When those bases are in position, along comes RNA polymerase, because we're making RNA, and RNA polymerase will form the phosphodiester bonds between bases that are next to each other, or nucleotides, between adjacent nucleotides, giving us a structure which we will refer to as pre-mRNA. We call this pre-messenger RNA, mRNA, because this region would be our exon, which we do want, but this region here might be our intron, which we don't want. The intron is the base sequence that's responsible for something else. It's not gene expression. It's not part of the base sequence for that protein. So before we can take our mRNA out, we need to get rid of the introns. And that is the job of these free-floating nucleotides that are also, the splicing enzymes that are also in the cytoplasm. So remember, the nucleoplasm has got all the stuff in it, that everything for all the processes to happen. So the nucleoplasm has in it our enzymes, has a free-floating nucleotide, also has our splicing enzymes in there as well. That splicing enzymes will come along and they will break the phosphodiester bond, removing the intron, giving us just the exon. Now we have mRNA. This mRNA, which is this region here, can now move out of the nuclear pore to the ribosomes. Now, these ribosomes might be on the rough endoplasmic reticulum, they might be in the cytoplasm. The base sequence is very specific. It matches now, it's complementary to the one that we had in the DNA. This base sequence is read in sets of threes. It's read in triplets. So the first three, UAU, is read like this. That's a codon. Okay, this is the next codon. If we look at the first codon, it's UAU. In the cytoplasm, we have tRNA molecules that have on them anticodons. These anticodons are complementary to the codons. So if we're looking at the base sequence UAU, the codon ATA will be complementary to it, and this tRNA molecule will move and make hydrogen bonds between the codon and anticodon. Okay, it will just position itself there. And what it does temporarily is just put this tyrosine where it needs to go. It positions that tyrosine in that position. The next codon is read, and the matching anticodon brings along its amino acid into that position, giving us alanine here. So we've got a sequence of amino acids as a result of the sequence of bases. This particular sequence, this very specific sequence of amino acids, gives us our primary structure of our protein, where you get this peptide bond being formed. You get secondary structure, which is our alpha coil and our beta-pleated sheet. We have our tertiary structure, which is our further folding, to give us our unique 3D structure, which is where insulin ends. Okay, now insulin can then, once it's made, be placed into a vesicle. The vesicle can then move towards the cell membrane. Remember the cell membrane, phospholipid bilayer. Okay, this membrane can fuse with this membrane, releasing its contents, so that insulin can go to where it's got to go to. Okay, this is just an example of insulin. This could be any protein, really, any protein. Okay, the process by which the mRNA is translated into a sequence of amino acids is called translation. Once the amino acid is in position, the tRNA molecule goes back into the cytoplasm to look for or to attach with another tyrosine molecule to bring it into position the next time. Okay. So we've got transcription in the nucleus, translation in the cytoplasm. We've got our helicase, our RNA polymerase, our splicing enzymes. We've got our free-floating nucleotides. All of these are in the nucleoplasm. We've got our small mRNA being made. Once the intron has been spliced out, it leaves, goes to the ribosome, where translation can take place.